And okay, so welcome, Anna. Anna Devor is an associate professor in the College of Engineering at Boston University. Prior to her position at BU, Professor Devor completed her PhD at the Hebrew University in Jerusalem and a postdoc in the optics group at the Martinez Center in Boston. She then directed the Neurovascular Imaging Laboratory at UC San Diego, as well as the UCSD two-photon imaging facility and the UCSD Center for Multiscale Imaging of Brain Function. Her research interests include cellular and systems-level neuroscience, as well as microscopy and the physiological underpinnings of non-invasive imaging methods. And today she's going to be talking to us about layer-resolved imaging of resting CMRO2 in awake mice with phosphorescent O2 probes. So thank you so much for joining us, Anna.
Sure, thank you, Tyler. So I have 1 million questions for Serge, and my brain is just about to explode. So let's see if I can focus on my own talk and kind of keep those questions for later. Okay, so I want to tell you about a recent study that we did using this, let me grab my pointer. Using this phosphorescent oxygen probes and looking at CMRO2 across layers in a mouse. In our case, all that we do is in mice. So to start with, I would like to talk a little bit about this overview of the world from our perspective. In our lab, we develop and apply tools, which are microscopic tools, mostly optical, but also some transparent electrodes. And we apply these tools to try to understand how these microscopic bits and pieces translate into larger scale macroscopic signals that can be detected non-invasively. So on the microscopic scale, again, everything is done in a mouse. We have these neuronal circuits, and we can think of this as these different populations of neurons. Maybe some of them are excitatory and some of them are inhibitory. And their activity drives in parallel, so that's a keyword, in parallel, vasodilation and oxygen consumption. We and others have done a series of studies that sort of support this idea that these processes really are parallel. And the first one is driven by feed-forward signaling. And the second one simply has to do with how much oxygen is needed. So this is in contrast to some earlier ideas where people thought that really it's the byproducts of energy metabolism that drive vasodilation. So we don't think that this is the case. Just to get it out of our way, we think that this may be the case, likely to be the case in different pathologies, but not under normal conditions. All right, so in our view, these are two parallel processes. And so now what we can do is that we can measure this activity using all the invasive tools available in animals. We can also use optogenetics so that we can try to manipulate the circuits. And then we can measure vasodilation as kind of precise as single vessel dilation and constriction that we can achieve with two-photon microscopy. And we can also look at oxygen availability and oxygen consumption using these probes. They are optical phosphorescence lifetime probes that we can use for that experiment. And so then at the end, with all those measurements, we can build those models that are embedded in realistic vasculature. So think of this as a voxel for our MRI measurement. And then we can predict those signals feed forward, okay? So this is not talking about inverse problems, just talking about the forward problem. So now in functional MRI, we know this well that we have these two processes that contribute to our bold response. So one of this is CBF, which is kind of the macroscopic counterpart of our microscopic dilation and constriction. And the other one is macroscopic CMRO2. And so these two, as we all know, they contribute to the bold response, but in opposite ways. And so this is kind of the fundamental uncertainty that we have in there. And this is why it's also so important to get handle on each one of those processes. And something that I don't want to talk about this today, but just to mention that one can also do similar kind of across species, across scale translation for actually electrical brain activity signals from neuronal circuits to this more macroscopic generation of electrical dipole that can be picked up by EEG and imaging. So just one word about the motivation for the study that I want to talk about right now. And this has to do with interpretation of the resting state functional MRI. So now resting state functional MRI is obviously a very popular signal that currently is used by kind of like everybody and their brother, including for those studies that are kind of clinically motivated. However, there's very little understanding what that is. And so we kind of, to address this, try to understand behavior of these microscopic parameters without stimulus, okay, just kind of under spontaneous neuronal activity conditions. And also, just to mention, we have this project that we call ourselves USA Rhythms team. So this is one of this Brain Initiative team projects. So here's John, here's Sheen, and obviously Bruce Rosen, Joe Mandeville. You probably know some other people, so David Kleinfeld isn't here. And also we have an amazing group of trainees. So here's Laura Lewis that you probably know. And so we're very fortunate to have all those amazing people working on kind of from different sides on this problem of trying to understand the resting state functional MRI on a more kind of biophysical basis. Okay, so there are lots of different measurements that kind of like all can fit into a single bottom-up model, but I just want to focus right now on one particular study. And this again has to do with two-photon oxygen imaging for the sake of estimation of cerebral metabolic rate of oxygen, CMRO2, across layers. So we're using this technology, which is called two-photon phosphorescence lifetime microscopy. We call it 2PLM. It's a flavor of two-photon microscopy. It's a lifetime imaging. So it's kind of like a case where we need to image decays and phosphorescent decays and reconstruct the lifetime. These measurements offer partial pressure of oxygen in TOR, on milliTOR or microTOR. And from this we can get this landscapes of PO2 in cerebral cortex in vivo, or in any other tissue that we are interested in. So now this allows quantification of oxygen metabolism. However, this quantification is model dependent, as we'll see in the moment. And this helps us understanding energetic costs of neuronal activity in general. And also obviously to look at kind of like laminarly resolved CMRO2, because it's a microscopic technology, so our view is very, very kind of microscopic. We published a few years ago, we published kind of a simulation environment that we're using in here to test our results. And the main result that I'm showing right now actually just has been published in PLOS Biology. So as we all know, and Luca and Serge and both Luca and Serge just did a very good kind of introduction and summarizing the idea of cortical layers. So cerebral cortex is composed of layers. So here is from a recent paper from Allen Institute, and here there's a reconstructed cortical column. So you can see that there are some differences in terms of layers, in terms of kind of what cell bodies are present in this particular layers. And also there's some vascular organization which is laminar as well. It's not as obvious. So this is a paper from David Kleinfeld's lab. And this is a quantification of that type of vascular reconstruction from Bruno Weber some years ago. And so I believe that this one was done in monkey. I also believe that this one was when Bruno was in Nikos Lagatidis' lab. And so in here, you can see that there are laminar differences. The red one, I believe, is penetrating arteries. So in terms of their length, there is not much difference. But when you get to the capillary bed, and so these green ones. So then obviously you can see that there is more capillary density in layer 4. There is also, we know there's also more cellular density in layer 4, because especially you know, in somatosensory cortex, but in other sensory cortices as well, the layer 4 is housing stellate cells, which are smaller and they're tighter packed. So cerebral cortex is composed of cortical layers. And now if we think in terms of laminarly resolved bold response, with the introduction that we just heard, there are two kind of questions that one can ask in terms of trying to understand whether it's reasonable to believe that there's going to be differences in the bold response across layers. So one has to do with vasculature. And in here, there's some, you know, there's some penetrating vessels that you can see in here. So these vessels, as we just heard, they penetrate all the way through, or most of them penetrate all the way through. And there's this, we know that the vascular walls can propagate dilation and constriction along the wall. And so this sort of brings a question of whether it's even kind of feasible to get some laminar differences, because it seems that whatever, you know, whatever kind of layer dilation starts in, it's going to be very soon propagated all across the column. And so there may be no difference at all. However, CMRO2 is local. And if you think of the cell, so here's this white one, for example, that has some dendrites and or axons. Mitochondria are located in all these processes, and they're very local. You know, there are even some synapses where there's very local mitochondria sitting at the synapse. And so there would be laminar sort of profile or some laminar resolution to CMRO2. So now, in terms of our imaging of oxygen, we use this phosphorescent probes, and this is the first one. So currently we have a second generation of this probe. It has been developed by Sergey Vinogradov from UPenn, and this kind of really revolutionized our way of measuring oxygen on this microscopic scale. So this work, we are working on this already for, you know, over a decade with Sava Zakadzik and David Boas. Sava is at Martinez, and David, as well as myself, we're currently at BU. So now I already told you that it's a phosphorescent probe. What it means is that there's a phosphorescent decay, there's a laser excitation pulse. We use pulsed lasers, so there's a pulse about maybe like 50 femtosecond or maybe like 100 femtosecond. And then there's going to be on a microsecond scale, there's going to be a decay. So, you know, the probe gets excited, then at some point it emits this phosphorescent photon. It's so-called forbidden transition on kind of quantum mechanic level. And so as a result of this, it takes longer time. And so these probes are relatively slow comparing to fluorescence. Fluorescence is in nanosecond, the lifetime of fluorescence. So in this case, it's in microseconds, so orders of magnitude. Okay, so a little bit slow, but it's like totally gorgeous. So you can see that this is a reconstruction of a piece of capillary bed, which is in between artery and a vein. So there's a little venule and there's a little arterial, and we can basically get oxygenation of every single capillary and get this awesome looking pictures. The other thing that we can do is that we can inject this probe into the tissue. So now it's going to sit not inside the cells, but in between cells in the interstitial space. And so now we are some few hundred microns below the surface, and we can see the surface vessels as shadows in here. There's another fluorescent dye inside the vessels, so we only see those that sort of cut through the plane that we're looking at. And in here, we're placing this grid of points, these are measurement points. And then using this phosphorescence lifetime, we know that, we measure the decay, and there's basically a lookup table between how fast is the decay and the actual PO2 value. And so we get those measurements in, in actual units of oxygen pressure. We have been working with Bruker to put this technology on their platform. Probably not very interesting for you guys, but just to mention that I have no financial interest in Bruker. So now, for years and years and years, we have been doing various measurements, including this tissue PO2 measurements under anesthesia. But then at some point, we kind of discovered that this is very, very bad. And the way that we actually discovered that this was so bad for us was in one of our previous oxygen imaging studies, where we reported, Sava and I, so we reported that about half of extracted oxygen actually get extracted from penetrating arteries in the cortex. And we thought that this is, somewhat unexpected observation, but we published this because that's how the data kind of work. And we were absolutely right. So this is what happened under our conditions. However, we did not know at that time that under anesthesia, our flow was very slow. And so because of slow kind of baseline flow, we were getting a lot of extraction already at the level of penetrating arterioles. And in an awake mouse, that is not the case. So as soon as we started doing awake mice, we were amused and amazed to see how fast the flow actually was. And this sort of was a lesson for us to learn that never do anesthesia again. So we currently are not doing anesthesia. We're doing all our experiments in awake mice, and also currently we translate into behaving mice. That makes experiments so much more difficult, but hopefully more translatable to what we actually care about. So now this is our phosphorescent microscope. So let's not worry about the details. This is, again, we're looking at the decay. So our mouse is awake. So here is a detail that may be worth mentioning. We have those chronic windows, right? Because we need optical transparency. Normally, skull is not transparent enough for us for two-photon imaging. But also we need to deliver this probe because there are lots of probes out there that are genetically encoded. So this one is not that way. So this probe actually needs to be micro-injected into the tissue to be able to do imaging. So what we do is that we have this rubber gasket in there, and there's this pipette, which is made of quartz. So it doesn't break easily. It can be pulled very, very thin. And we go in and we basically deliver this probe maybe like an hour before we want to measure. Okay, so now what do we do next? Well, so we also inject IV, a little bit of fluorescent tracer that stays inside the vasculature. And so now this is what we see. So we see there is an artery in here, and here it penetrates, goes down. And then we can trace it. So we keep going deeper and deeper and deeper, and we can see this sort of, making its way down. And then at a certain plane that we like, we can do our measurements and then try to extract CMRO2 in a way that I will show you in a second. Okay. So how do we do this? Well, this is the fluorescent tracer. Let's say that we're at this plane. Again, this is a super microscopic, resolution. So this is 50 microns, this bar in here. And then we put a measurement grid. And then we have for each one of those points, we will have a photon count, and we also will have our PO2 map. The good thing about lifetime fluorescent or phosphorescent is that our measurements do not depend on brightness. So this actually in here, I'm showing this photon count to show you that the vessel overshadows itself as it dives down because it's diving a little bit at an angle. So we can see the shadow. However, when we get to the PO2 measurement, because it's lifetime, it's kind of independent of photon count or intensity. And so we're getting these maps, which look very different from the actual maps of photon count. So now we have to excite multiple times to build this histogram. This is a histogram, basically, of probability of us detecting a photon after a pulse, a laser pulse. And then after we do this, we kind of look at our residuals from the fit, this fit of the decay of the phosphorescence. And then we figure out how many kind of excitation cycles we need. And this basically determines our speed. So in here, you can see that our error is going to be, pretty high if there are very few cycles. By the time we get to maybe like 1,000 cycles, our error more or less, asymptotes. And so we usually use this 1,000 cycles. And so this 1,000 cycles for this 400 points in here, it basically tells us that it takes us about 2.5 minutes to acquire a grid like that. All right, so now what do we do with this grid? Well, so Sava and I published this paper back in 2016. And so at that time, we basically looked at vascular organization and paid attention to this very interesting feature. And the feature is that if you look at diving arteries from top, okay, so this is a two-photon vascular stack, and we're looking from top down. You'll see that there is this void of capillaries around diving arteries. I would say diving arterioles, they're small arterioles. And the reason for this probably is just simply because during angiogenesis, when the vessels grow, they sense oxygen. And because there's so much oxygen leaking from these vessels, there's not much capillary density around them. And as a result of this, we kind of can fool ourselves into believing that that we can apply this Krog model of oxygen diffusion from a single cylinder, which is a huge simplification, but it allows us to estimate CMRO2. And there are a few parameters in there, so we need to know the intra-arterial PO2. We kind of need to know where exactly we are within this radius of void. And then we can feed certain formalism to this, and we can, so this is our measurement, and this is the CMRO2 that we want to get. This is diffusivity and solubility of oxygen. This is our radius of the artery, and this is the radius where we are, so kind of like anywhere in here. The other thing to mention is that if we really look at higher resolution imaging of PO2 inside the vessel, you also can see that it's leaving from the edge, right? So there's higher PO2 in the middle, and there is lower PO2 in kind of at the wall in here, and this is because some of this is living into the tissue. So this is from that paper. Sorry, I'm moving my Zoom window because it's not where I need it to be. Okay, got rid of it. So this is from that study that we published in 2016. And what we did in here is that we placed a grid like this. There was a diving arterial, and here we used different level of anesthesia. And so when the anesthesia was very deep, we could see that this profile was opening up. And the reason for this is that when the anesthesia is deep, there is less consumption. So this kind of profile from the center of the vessel into the tissue starts opening up because there's no kind of consumption in the tissue that pulls it down. Recently, we updated this model a little bit, so we kind of got a better fitting. I don't want to go too much into details, but this is again a published paper. We can also talk more about this during discussion. But the basic idea of the model is the same. It's a modified Krog model of diffusion from a single cylinder. And so this again is the same idea. We basically go down, we follow diving arterials, then we place our grids, and then we look at this profile. So here's now this profile, and we estimate CMRO2 based on that model. The vessel is here, so this is the 0 here, so that's the vessel wall, and this is the radius into the tissue. And so we can see that there is this kind of, there is this gradient around, and we can fit this gradient to get our CMRO2. And now we can repeat it in different depths, right? So we just go and we do this in different layers. And so here's a result, okay? So the M is for mouse. So this is 8 mice here. And now this is the cortical depth, and this is the CMRO2. And going from the surface to 500 microns down, we have a decrease in CMRO2 at the baseline, right, during resting state. A couple of points that I would like to make to clarify in here. So one of them is that this model obviously assumes this central cylindrical symmetry. When we go to the actual biology, it does not quite look that way. Why? Well, because there's some extra vessels around. So what we do is that we have to segment this. We have to segment this measurement grid such that, you know, this higher oxygenation, which happens there due to like this vessel, for example, or in here due to this vessel in here, or not, you know, or just not skewing our estimation of CMRO2. And this is where we also used the simulation environment that we published a couple of years ago in neurophotonics. And what we did in there is that we just used synthetic data, and where we kind of knew what exactly is the CMRO2 because we put it into the model, and then we placed some vessels in different locations and tried to recover CMRO2 using our data analysis stream. And so in here, this is what we do. So we basically are using those tools that have been validated to the extent that we can within this synthetic environment. Okay, so now when we started this study, my expectation was that the CMRO2 is going to be highest in layer 4. Layer 4 in a mouse is already at 400 microns. So 400 microns in here, this is layer 4. And at 500, it's already layer 5. There are very few points, 2 out of those 8 mice that we could achieve at 500. And the reason for this is that 2-photon microscopy can penetrate through hundreds of microns, but it depends on the density of the probe. And as denser the staining gets, as more difficult it is to penetrate, because we start getting so-called out of focus phosphorescence in this case. And so in this case, the probe is super dense because it's basically everywhere in the interstitial medium. So it's tough for us to penetrate. We did some controls for this, and we can talk more about this. We don't think that these measurements kind of are, we think that these measurements are good. But we had to exclude most of them at 500, and we could not use any of our deeper measurements because our out of focus signal was growing up, meaning that the measurement is not local anymore. So yeah. So now the other thing that I would like to point out in here is that, you know, so it's beautiful that we have such kind of ability to look at such high resolution, but also it limits us in a way that our vision is super microscopic. So what we're doing in here is that we estimate the CMRO2 based on this region of maybe like 200 microns around diving artery, but then we make this assumption that we would like to believe that is true. But I understand if you guys become critical of it, and the assumption is that we have basically space invariant CMRO2 within the layer, within the same depth, at least at the scale of like a single cortical column. So this is one of those controls that that has to do with out of focus fluorescence. I'm going to skip it for you because I don't think that this is of much interest to you. And I want to go through these conclusions and then very briefly show you something else. So to conclude, this measurements basically show that during the spontaneous activity, which is called resting state, but kind of historically, we have been calling this just spontaneous neuronal activity. The highest CMRO2 is found in cortical area 1. Well, this is what our measurements show. So we don't know why. One possibility is that we're talking about resting state, right? So there's no stimulus. Layer 4 is really reserved for, let's say in whisker barrel cortex, it's reserved for inputs from thalamus, right? And if there's no stimulus, there's no input from thalamus. So during, when there's no stimulus, layer 4 actually is nice and quiet. However, there's lots of cortical interactions that happen in other layers, and chiefly in layer 1. Because internally generated brain activity, meaning kind of like communication between different brain regions, are very busy in layer 1. So there's lots of inputs from one area to the other, and all of them synapse on this kind of apical tufts of pyramidal cells in layer 1. That's an interpretation, right? So that's not based on our measurement. That is a speculation of why this may be happening. So now the other interesting thing to consider at the speculation level is that the laminar CMRO2 profile is very likely to be a function of what kind of computation actually is performed, right? And so this is why we're currently working on combining this to PLM with other measurements that will give us this parameter of the brain state. And we have 2 of those. One of them is our transparent electrode arrays. We're working on this. We published some other papers on transparent electrode arrays. We haven't been we're still in infancy in combining this with 2PLM. And the other one is using optical probes for brain state. And so this together would allow addressing questions such as what is the energy cost of being alert or paying attention, in addition to all this kind of interesting questions of the physiological basis of the bold response. So now about just kind of one word really about the optical probes of neuromodulation. So currently we have, this probes just became available sort of like last 2 years. And these are genetically encoded probes, and they're available currently for acetylcholine, but also for dopamine and for serotonin and other things. And the way that we record them, we measure them, is we can do 2-photon imaging, but we also can do single-photon imaging. And the single-photon imaging is giving you really kind of large view of the entire part, the dorsal part of the mouse cortex. We can image them all together because there is some color multiplexing going on, so let me not bother you with that. But I'll just can show you kind of like an example of how this looks. So these are these 2 two hemispheres that we're looking from the top. And we can see, so this is just a movie of this ongoing activity. So this is the mouse twitching its nose. And so we also, can do some other measurements, such as the pupil of the eye. You can see that every now and again, it just becomes very large and then it goes small. So this has to do with the brain state. And also, this is the acetylcholine probe. We can image this in space and time. We can combine this with calcium imaging in, local cortical circuits. And also we can do optical oxydeoxy total hemoglobin. And so we're currently working on this. This is very much work in progress, but we're working on this, trying to kind of understand how this neuromodulation factors into our stimulus-induced and resting state, hemodynamic impulse response functions, things like that. Okay, so I just want to thank all those beautiful people that I have pleasure and privilege to work with. And I just want to plug this in. We have, I'm an editor-in-chief in Neurophotonics, which is, it's an optical journal, but it just published these 2 papers that may be of interest to you. And they are papers with lots and lots of authors, and they're all, brilliant people. So one of them has to do with microscopy. We call them status reports. So this is the tools for microscopic measurements. And this is including hemodynamics, but also like anything else, like those probes that I just talked about, or, some other measurements. And the other one is the non-invasive counterpart of this, also optical. So those are fNIRS and DCS and things like that. And so I'll just stop here.
Thank you so much, Anna.